Good morning, everyone. So today's topic is role of thrombin receptor mediated platelet activation. Don't have much first-hand experience on this subject, but uh, we will together see what we can understand. So the outline of this presentation would be very brief about platelets and thrombin activable receptors, lab methods by which we can assess the platelet functions, the role of platelet activation and the aggregation in cirrhosis based on recent research of last 3-4 years and their contrasting views, and overall overview of various platelet dysfunction which can occur cirrhosis and its complications, and limited experience in ILBS and way forward. So I'll start with the first brief introduction. So the platelets, they play a role in hemostasis and thrombosis along with the maintenance of the vascular tone, inflammatory infiltrate, host defense mechanism, tumor biology, and also recently they are responsible for the bone marrow hematopoietic stem cell maintenance and their maturation. In terms of hemostasis and thrombosis, the platelet primary role is primary hemostasis, that is a weak platelet plug formation, the initial hemostasis formation, and which depends upon the three things, that is the platelet count, platelet functions, and the von Willebrand factor. Now, during this primary plug formation, there will be platelet adhesion, their activation, and aggregation. So these three things are not separate altogether. They are interplay between these three things together. And finally, there will be a weak plug formation upon which the cascading of the coagulation system will take place, and a stable clot will form. And finally, after that, there will be a clot resolution by fibrinolysis. So now this is the diagrammatic presentation. So please pay attention to this diagram. So first, this is the vascular endothelium, and this is the subendothelial matrix. When there is an injury, it is exposed, and through the von Willebrand factor and the platelet receptor GP1B, there will be interaction alongside with the collagen and GP6. So these two receptors of the platelets, they will interact first hand with the exposed endothelium. And after that, there will be a small surge of the thrombin. It is not a thrombin generation as such. It is a small amount of thrombin is generated to initiate the action. So this thrombin will attach to the receptor PAR1 and PAR4. We will discuss PAR1 and PAR4 subsequently. And then there will be the alkaline environment by the calcium surge, and subsequently there will be the release of dense granules, alpha granules, and the alpha granules they will secrete ADP, and ADP will does two things. It is responsible for pseudopodia formation, that is for the platelet activation and the cascading effect of the coagulation. Thromboxane is also released, which is responsible for the vasoconstriction and platelet aggregation. And they also, this thrombin, by the interaction with its receptor, will leads to the activation of the platelet. The activation is assessed by two things. One is the platelet interacting receptor expression, that is GP2B or 3A, and the P-selectin expression. So this is overall activation, and we cannot segregate platelet activation with aggregation as such. Adhesion, aggregation, and activation, these three things are all together. And then there will be, through the P-selectin, there will be the other cells which will interact with the clot, and the coagulation cascade will take place. So leave apart this complex thing. Now we come to the thrombin activable receptors which are there on the platelets. There are several receptors, but for the platelet, the thrombin by which the receptors are activated are PAR1 and PAR4. The basic difference between PAR1 and PAR4 is PAR1 is a sensitive receptor which will be activated by the small concentration of thrombin, and this action is transient, so it will be relieved briskly. The PAR4, it requires a lot of thrombin concentration, not lot of, relatively larger amount of the thrombin concentration, and the effect will remain for prolonged period, and it is a potent action. So this is the difference between PAR1 and PAR4. So when the platelets are activated, both receptors are responsible for it, and this PAR1 is a primary receptor in human beings, whereas the PAR4 is in animals. PAR2 is not related to coagulation, it is related to inflammation and the cytokine secretion. So lab method by which we can assess the platelet functions, it is summarized in this table. There is no laboratory test that can assess the vessel wall or endothelial, which is the foremost part of the coagulation system to take place. And these systems, they have a limitation that they analyze separately the various parts, that is some part of platelet activation and then aggregation or the granule content. So there is no method by which we can encompass all three things together, and there is that's why there is no pivotal screening test available for the platelet functions by the lab methods. So what lab methods we have? Initially we used to do BT, that is the bleeding time, which is obsolete now. The finer version of the bleeding time is the platelet function analyzer, which is clinically used for the platelet antagonist usage, and it is rapid, automated. Then this is light transmission aggregometry. This is done by the platelet-rich plasma. So this is the reliable reference method up till now. And but the problem with this is there are a lot of pre-analytical steps are there, and which has to be standardized, and it is quite sensitive to it. Impedance aggregometry, we will come to those things later, but just for the understanding briefly, impedance aggregometry we can do on whole blood, and it is also rapid, but it is sensitive to the platelet count. So all those methods which we have discussed, particularly the PFA, the light transmission aggregometry, and impedance aggregometry, these three things can assess the platelet function, but they are sensitive for the platelet count. So we have to have more than 100,000 platelet count to assess adequately platelet functions. So either we have to adjust according to the platelet count, or we can say that we can reliably do on those patients, not in cirrhotic patients especially. So how we will do in cirrhotic patient, we have to adjust according to the platelets. So these are the three methods by which we can assess. Recently, flow cytometry method is being used to assess the platelet activation. But flow cytometry method, we will come about the limitation of it. So what the recent article says about the role of platelet activation, aggregation in cirrhosis based on recent research, and there are contrasting views. So first we will discuss that this is decreased, meaning platelet activation and aggregation will be decreased in cirrhotic patients. So this is a study a few years ago which suggested that when you put up the agonist to activate the platelet, the activation markers which are there, the P-selectin and others, they will be reduced in case of the liver cirrhosis as compared to healthy control. This is platelet activation. And when you do the aggregation, it also says that the aggregation in case of the cirrhosis, it will be reduced as compared to healthy control. So the conclusion of this slide is basically in cirrhotic patients, both the activation of the platelet is reduced and the aggregation is also reduced. Now, the another study which also says the similar thing, that in case of cirrhotic patients, the aggregation by various agonists, the agonist for this restocitane, ADP, collagen, epinephrine, D-dimer, these can be agonist, so they can use to activate the platelet and subsequently the aggregation. So they are reduced in cirrhosis as compared to control, and as the disease progresses from child A to child C, the platelet function or the aggregation will also reduce. Further, when we compare the compensated cirrhosis versus decompensated cirrhosis without ascites and with ascites, again the platelet aggregation with various agonists will reduce as compared to the controls or the compensated liver disease. Now, this is the article which was recently published in hepatology. So, what they did is they took 100 odd patients and they assessed the platelet activation and the role of platelet activation, possibly which could be on bleeding or thrombotic events, risk of hepatic decompensation, or transplant-free survival. So the receptor we have discussed is PAR 1 and PAR 4. So they put agonist to these receptors, and subsequently when the platelet gets activated, they will express P-selectin and activated glycoprotein 2B. So they assessed two markers, P-selectin and glycoprotein 2B. Now, the summary of the initial summary of the findings is these activated molecules, they were significantly lower in case of cirrhosis by both agonist as compared to control. Means the healthy patients, they were lesser. And there was also inverse correlation with the liver disease severity. It means when the disease progresses, the platelet activation will also goes down. And when they assess with the pro-inflammatory markers, it showed a positive correlation. That means that, negative correlation. That means when there is a infection, inflammation scenario in case of the milieu, cirrhotic milieu, the platelet activation will get reduced. And when you compare amongst the etiology, the ALD patients, they showed the significant reduction in the platelet activation, whereas the cases of NASH or MAFLD, they showed increased platelet activation as compared to the other etiologies. Now, whether it could be able to predict bleeding or thrombosis in those patients, so they followed up those patients with the median interval of 25 odd months, and the bleeding events were in 10% of the patients, and most of them were related to GI bleeding, the variceal bleeding because of the portal hypertension. So as expected, the HVPG was the predictor of the bleeding, but not the platelet activation markers. They were not indicating that it will predict the bleeding episode. Similarly, the non-tumoral thrombosis that appeared in 7 patients, and most of them were portal vein thrombosis, and they found that the epinephrine induced P-selectin can help in prediction of the development of PVT. This is non-tumoral overall. Most of them were PVT, so similar results are there. So the epinephrine-induced platelet activation, it itself suggests that the platelets are activated and they can lead to thrombosis. So the activation reduction is little overinterpreted here. So now the prediction of hepatic decompensation, they developed various multivariate models, and they showed that the hepatic decompensation can be predicted by the reduction in the platelet activation. P-selectin activation P-selectin expression and GP3 2B 3A can. Epinephrine is put as an agonist, and that will cause the release of the.
It is on the in vitro.
Yes, sir, in vitro test. So that will cause the release of the granules in the platelets, and that will cause the platelet activation. It is suggested if it is there, then it is marker of thrombosis. So whether it is a predictor of hepatic decompensation, the second objective of their study, they showed some of the markers can say that lower platelet activation can predict the hepatic decompensation. But there were a lot of models were applied, and then only they were able to say something about this. And finally, they have to dichotomize the values, the markers, P-selectin and the glycoprotein. So based on the dichotomization, they could say that hepatic decompensation can be predicted by the lower level of platelet P-selectin and glycoprotein 2B3A. This is about hepatic decompensation, which appeared in 29 patients, and transplant-free survival also predicted by that. Similar group also studied the another and they published in thrombosis and hemostasis in 2023. The findings are similar. It says that they took the cirrhotic patients with the clinically significant portal hypertension. 100 odd patients were again taken, and platelet counts were similar across the child categories. The expression of these platelet activation marker was significantly lower in case of child B and C as compared to child A, and these markers were also reduced further reduced in case of child C. And the P-selectin and activated glycoprotein, they were lower when the portal hypertension was prominent, like HPPG when it was more than 20. So the conclusion of this study is that the platelet activation marker, they goes down when there is an advanced liver disease and there is portal hypertension, advanced portal hypertension. Now limitation, both these studies, they were done by the flow cytometry, by the agonist, but there are several limitations in this study. Number one is the flow cytometry, which is not standardized for the platelet function as of now. They use the suboptimal concentration of the agonist, and they did not use the trap thrombin agonist, which is the real agonist for the platelet receptors. So they use something else by the Roche, and it was not used, and it was also used in suboptimal concentration. The event prediction was only significant after the dichotomization of the markers, that is the P-selectin and glycoprotein. And when you see this figure, this GP glycoprotein 2B, it also takes the advantage of fibrinogen to get activated and for its function and for its activity. So they have not considered this study for the glycoprotein 2, and it was not adjusted for it. And the event number were very less, especially those related to the hemostasis, which is the bleeding or thrombosis. The similar group again studied the platelet aggregation by the PFA 100, and they took 700 odd patients, and they found that the closure time, that is the platelet dysfunction basically, it increases with the CTP, with the MELD, and with the portal hypertension, but it is not related with the hepatic decompensation, or it is not a predictor of hepatic decompensation or liver-related mortality. And also they have concluded that the traditional markers, HVPG and MELDs, are much better than the platelet markers or platelet aggregometry over the period of 5 years they did the AUC, and they found every time that HUPG and MELD are much better than the platelet function aggregometry. So now, what we understood till now, it says that platelet function or platelet activation or platelet aggregation, they decreases with the advancement of the cirrhosis and with the portal hypertension. So overall, because of the milieu, nitric oxide, fibrinogen degradation product, and ApoE, they will reduce the platelet activity, and the platelets in cirrhosis, they have 5 times lesser response to this phospholipase C, and it causes the impaired mobilization of calcium inside the cytoplasm, so the platelet activity will be lesser, and the alpha and the dense granules are less in case of cirrhosis. The synthesis of the thromboxane A is defective. The layer, that is the phospholipid layer of the platelet, is altered. So overall, the platelets are less activated or less functional or less aggregation type of platelets are in cirrhosis. But there are several other things which can get them activated, like the circulating LPS or subclinical toxemia and the increased expression of P-selectin. So these things can, so we understood till now that the studies says the platelet function or platelet activation is decreased in cirrhosis. And it was suggested that platelets may be activated initially, they remain in circulation, they lose their granules and become less activable or functional, and that is called platelet exhaustion. But the enigma will not end here. What we see in under the microscopy, so we will take one by one. This is the normal platelet, which is granular, small, and this is the agranular platelet, and this is the hypogranular platelet, and this is giant platelet. So in case of cirrhosis, we don't see too many of agranular or hypogranular platelet. It means they are having granules. So they may be functional, but the results is overinterpreted that they are exhausted. In some cases, yes, that could be, but not in all. Now the contrasting view because of this is the platelet aggregation is increased in case of cirrhosis. So this is another recent study where they they took 200 patients of cirrhosis and 60 chronic hepatitis and 40 healthy subjects, and they did the platelet aggregometry, but they adjusted it for the platelet count, absolute platelet count, and they call this ratio as PLT ratio or platelet ratio. In absolute aggregation, if you take the absolute value, that is the AUC, which may be decreased in cirrhosis and chronic hepatitis as compared to control, but when we adjust according to the platelet count, they suggested that the platelet ratio, means the aggregation of the platelet, is increased in cirrhosis and chronic hepatitis than the controls. Further, this ratio is higher in case of alcoholics as compared to non-alcoholics. We saw in previous study that the platelet activity is lesser in case of the alcoholic patients. On subgroup comparison of the alcoholic patients, the recent drinkers or the abstinence of 1 month, there was no significant difference was there. And also, if we take a cutoff as a median PLT ratio of the normal individuals, those with the advanced liver disease, they have higher ratio as compared to, or those patients are in the higher category. Hope you would understand this. That means if the value is 5, then child C patient, most of the, like 87% of the patients will have the higher PLT ratio than the normal individuals. And this PLT ratio is also higher as we go by the advancement of the disease. So in child C, the aggregation parameter is much more than the compensated disease or chronic hepatitis or child A. Moreover, even in case of thrombocytopenia, there is a platelet count is less in number, even if then the aggregation is much more potent in cirrhotic patients. And subsequently, they utilize this for the clinical application, like whether it will be able to predict further decompensation of the cirrhosis or the liver transplantation or the liver-related death. And they found that the PLT ratio, those who had the further decompensation, they have higher PLT ratio as compared to those who did not. And also on multivariate analysis, the platelet ratio was the significant predictor of this hepatic decompensation and liver transplantation and liver-related deaths. And after triaging this into 3 categories, they suggested that the platelet aggregation parameter, if it is more, then there are more chances of the hepatic decompensation or liver-related deaths. Another study where they studied 128 patients of the decompensated cirrhosis. They followed up for 12 months, and 18 of them developed PVT. So the PVT development can be predicted by the PLT ratio. That means the aggregation is more in cirrhosis, and they can be able to predict the thrombotic episode or event in case of cirrhosis. Patients who are undergoing transplant, this is another study which showed that the platelet aggregation in case of cirrhosis is much more, and when you adjusted for the platelet count as well, it is higher as compared to the controls. And this is with the various agonists. You can see the aggregation is prominent in case of cirrhosis as compared to control, and over the period of the time, this aggregation parameter will normalize in case of the liver transplant patients, and this will normalize within 28 days. The problem is the platelet aggregation or aggregation ratio is that, please look at this diagram here. This is the impedance done by the impedance aggregometry. So whenever there is a agonist, the platelet will stick to the electrodes. But in case of cirrhosis, because the platelet volume is or mean platelet volume is much higher than the control or the other diseases, so we expect that the heavy platelet or larger platelet will cause the more impedance when you put across the 2 electrodes. So it does not necessarily be because of the platelet aggregation. It may be because of the volume, because volume will occupy this space and it will cause the impedance. So this is the problem, that this is also over-interpreted. Now the clinical application, this is a large cohort study and of cirrhosis, 6,000 cirrhotic patients compared to the 24,000 matched comparators, and they showed the cirrhosis patients, decompensation of the cirrhosis will increase the risk of acute myocardial infarction, whereas the cirrhotic patients, they also have more episodes of the veno-occlusive disease or thromboembolism. The animal study which suggested that the platelets in case of NASH, they will go inside the liver and they activate the Kupffer cells and subsequently the various inflammatory cells and causes the exaggeration or the progression of the NASH or HCC development in case of the animal model. So overall, clinical investigation, it discovers the basic altered hemostasis in liver disease, but basic researchers, they identified that there is a mechanistic link that the hemostatic system, that is the primary hemostatic system, can cause the liver toxicity and progression of the disease. and we need to have a crossover of these findings. Balanced view is that a larger study was conducted with all possible parameters of the coagulation system was taken along with the clinical parameters, and they found that portal vein thrombosis development is dependent upon the portal blood flow. I don't know the full form, the hepatologist would know. The portal blood flow volume and the variceal bleeding, then the all coagulation parameters. Whatever can be done in coagulation system, they did all those tests, but identified that the portal vein thrombosis, it depends upon the clinical parameter. This is the comprehensive view where expect that there will be platelet dysfunction in case of cirrhosis, and because of that there will be lot of circulating markers will be increased, like the thromboglobulin or platelet factor 4 or p-selectin. So in circulation we can identify those markers, and they suggested that their platelets are activated because the volume of these markers were much higher in cirrhotic patient. But the issue is that they are cleared by the liver. So if the liver is not functioning well, so they will not be cleared, they will remain in the circulation. So you will say that the platelet activation markers are higher in case of cirrhosis. But overall, when you see the endogenous thrombin generation potential, whether it is in case of control or child A, B, C, you will see the absolute thrombin generation is almost similar, whatever the situation is. So this is a more balanced view in case of cirrhosis. Now coming to the last few slides, the etiology and complication specific platelet dysfunction. The alcohol, it has an inhibitory effect on platelet function. However, all studies will not say the same. HBV, HCV related etiology can cause platelet activation. Cholestatic liver disease initially can cause the platelet activation, but subsequently because of the toxic bile acids, the platelet function will deteriorate. Sclerosing cholangitis, it increases the platelet adhesion, create a hypercoagulable status, but eventually it may disappear when the disease will advance. AKI and bacterial infection can impair the primary hemostasis and hemorrhagic risk. Anemia, of course, this requires a separate topic on this, how the anemia will affect the platelet functions. And HCC is associated with the platelet activation and aggregation. Why? I'm sorry about this slide. Because of lot of factors which are secreted by the HCC, so there can be hypercoagulable status, and we few years ago, these are three categories of the hepatocellular carcinoma. Group A, where there was a single mass in the liver. This is group 2, where the liver mass and satellite nodules inside the liver, and this is with the distant metastasis. So we saw that those when we do the FNAC of HCC, so we saw that those patients who have distant metastasis, their HCC will be wrapped by the platelets so that they can embolize or go to the distant sites. That's how the HCC can cause the pro or hypercoagulable status. Platelet function after transplantation, they will recover by the 28 days. And this is the ILBS data study published by Jassi, which says that hyperoxidized albumin can activate the platelet and subsequently the inflammatory conditions in case of alcoholic hepatitis. And recently we have acquired this light transmission aggregometer, where we are currently using the platelet function assessment by the ADP, collagen, epinephrine, and restocytin. And this is the patient data which we have done in 8 patients. Now coming to this patient particularly, the platelet count was 341, but with ADP the value was 4.5, collagen 11 point. It should be above 50, and epinephrine it was 2.5, and restocytin it was 22.2. So all the parameters were reduced in this patient, and this patient might be a case of von Willebrand disease uh, or Glanzmann thrombocythemia, even if he is having a normal platelet count. So we saw one thing where the platelets get activated and they lose granules because and the exhausted platelets. So this patient, the ADP, because of the ADP, the activity was less. That means this patient might be having the hypogranularity of cirrhosis, and that's why even if with the epinephrine it was quite okay, but with ADP, because it was not released, so the function was not optimum. Adjust it according to, we have to calculate in 100 platelets. So accordingly we have to multiply by the factor. Suppose the platelet count is 21,000, the whatever value are coming, we have to multiply by 5.
No.
Which patients? So
Yeah,
this
number
one?
4. No, below. Yeah,
This
one.
one.
No, above this.
Above there.
Yeah.
Okay. Sir, it was ADP 116, 64, 61 and 98. So it is okay.
It is okay.
Yes. Our own study. Because of the contradicting results, we need to undertake platelet function studies, which will be adjusted for the platelets and the MPP, the mean platelet volume. And flow cytometric studies can be considered with the adjustment to the fibrinogen and with the titrated concentration of agonist, not the suboptimal concentration which they used. So conclusion, basically there is no conclusion, contrasting views based on the methods, and no conclusion as of now can be drawn, and we need to validate ourselves and we need to do our own studies.